Hello, everyone who's watching right now or later. I'm just going to be going through this paper relatively quickly. So I'll leave out most of the sort of recall style questions. I'm going to be focusing more on the concept style questions. So if we go into the first question, immediately, so this, you can see an electron micrograph of a mitochondrion. Okay, state the function of mitochondria. So this one is a recall question. So mitochondria is the site of ATP production, or you could also mention aerobic respiration. Okay, structure A is one of the cristae. So these are sort of infoldings, invaginations of the membrane on which ATP production takes place. So cristae, or crista, singular. Okay. Calculate the magnification in the image in figure 1.1. Okay, so unfortunately I don't have a ruler with me at the moment, but it's around 35 millimeters or 34 millimeters. And the way you calculate the magnification is using this triangle. So if you, so if you have the image size on top, you have the actual size and the magnification. Then to calculate any of these three, you cover it up and then you get the formula. So in this case, we want the magnification. So to get that formula, that would be m equals I over A. And then if we measure it out, so 35 millimeters over 0.7 micrometers, but these are not in the same units. So we need to add 10 to the 3. So that would be to make these both in micrometers. And we would get the answer, which is 50,000, I think, times 50,000. Okay. Next, explain why the light microscope could not be used to produce the image in figure 1.1. Okay, so we can see that the scale bar, that's about half of the mitochondrion, is 0.7 micrometers. So the mitochondrion will be just over 1 micrometer across, and the maximum resolution that we can get with the light microscope is around 250 nanometers. So you would be able to see the mitochondrion with the light microscope, but the problem is that the wavelength of light is too short to give enough detail, to give enough resolution, to go into the level of detail that you see on this electron micrograph. So if you think about how thin the membrane is that we can see here, so that's about 7 nanometers across. So you wouldn't be able to see this much detail at all. Yeah, and that's just a property of visible light. That's, yeah, as short as you can go. Okay. Scientists think that mitochondria were once prokaryotes. The evidence for this is that mitochondria have features in common with prokaryotes. State 2 features that mitochondria have in common with prokaryotes. Okay, so this question's hinting at the endosymbiotic relationship between eukaryotic cells and mitochondria. So there's a number of different things that you could mention here. One, I think, which is the sort of one of the more obvious ones, is that it's got a circular DNA, so it's not packaged in the same way, in a linear way, like a eukaryotic cell's genes are. If you think about chromosomes versus the circular DNA. Another one that's, I think, an easy one to remember is the fact that mitochondria are just, in general, a similar size to prokaryotes, a similar size to bacteria. And another really important one is that they've got 70S ribosomes instead of 80S. So this one is almost undisputable, the fact that they use a completely different type of ribosome than their host cells suggests that they indeed do derive from prokaryotic origins. Okay. Figure 2.1 is a diagram of the structure of a protein channel for ions in a cell surface membrane. Figure 2.1a shows the channel when open, and figure 2.1b shows the same channel when closed. Okay. Name the process by which ions pass across the membrane using channel proteins. This one, another quick recall question, facilitated diffusion. So that means passive diffusion across the membrane through a protein channel, facilitated by a protein channel. Explain why a channel protein is needed for ions to pass across a cell membrane. So there's a number of things you can mention here. So one of the easy ones to go for is that if you imagine your standard phospholipid bilayer here, then you will see that indeed a phospholipid molecule has a hydrophobic tail and a hydrophilic head. So this is the phosphate, and these are the fatty acids. So if you think about an ion that wants to go through this, it's not going to have a really easy time going through this hydrophobic layer. So one of the ways in which this can be overcome is by using some sort of a channel which has a hydrophilic channel, hydrophilic part in the middle, sort of like a cylinder. It's through which anything that's water soluble or charged can go through. And the way that's achieved is that if you have a transmembrane protein, you will have in the middle of it hydrophilic R groups of amino acids that facilitate the ions passing through the membrane. Okay. The channel protein in figure 2.1 is made from 5 identical polypeptide chains. Name the level of protein structure which is present when 5 polypeptide chains form the protein. So polypeptide chains to polypeptide chains, that would be the quaternary structure. Okay, the part labeled C in figure 2.1 is another level of protein structure. Name this level. Okay, so C, we can see from this coil, that's a very characteristic alpha helix, which is an easy hint at the fact that this is referring to secondary structure. So if you recall secondary structure, normally what they mean is interactions between the carboxylic head group and the amino group, forming a beta sheet or an alpha helix. Channel proteins are examples of transmembrane proteins. The polypeptides are held together and also interact with phospholipids in the membrane. Suggest how the polypeptides are held together and suggest how they interact with phospholipids. Okay, so let's break this down to 2 parts. First, how the polypeptides are held together, and then how they interact with the phospholipids. Okay, so polypeptides are held together by intermolecular forces. You can name many examples of this. So hydrogen bonds, hydrophobic interactions would be a good one. disulfide bridges. So anything, if you just go through them individually, interactions between the R groups, those can be any of these mentioned. So the disulfide bridges, the ionic bonds, hydrophobic interactions, and of course the ultimate example that you can always pull out, van der Waals forces. And then how they interact with phospholipids. So again, going back to sort of the channel protein example, the way that a protein will be inserted into the membrane is that parts that are exposed to the hydrophobic part of the membrane will have hydrophobic amino acids so that they can interact with that part of the membrane. And then they will have hydrophilic charged groups, R groups, that interact with the phospholipid head. Okay. Figure 3.1, in a light micrograph of cells in the root tip of the garlic plant Allium sativum, it has a diploid number 2n of 16, okay? So 2n, 16, okay? Name the stage of mitosis shown in cell D. So this one's another recall one. We can see that the chromosomes have already been pulled to the poles of the cell. So that is anaphase. Late anaphase, namely. Explain why mitosis occurs in a plant such as A. sativum. So, number of different things that you can mention here. In response to wounding, you would need to repair damaged tissue, so you would need to replace cells that were wounded, or in addition to this, to replace dead cells. Also, an important one is growth. So the only way, so this is an obvious way in which you would produce more cells. And in addition to that, you could also be you could also have mitosis for asexual reproduction. Okay, state the haploid number of A. sativum. So if 2n is 16, then n is 8. Explain why a plant such as A. sativum produces haploid cells. So, as with any other eukaryotic organism, for sexual reproduction, to form gametes, so it will have its genetic makeup so that it can form zygotes. And furthermore, it also prevents a doubling of the chromosome number. Okay. DNA replication must occur before cell division. Describe the process of DNA replication. I think I won't spend too much time on this because this is literally asking for your textbook knowledge. Do you or do you not know how DNA replication occurs? So just to mention, just quickly go through the key points that you need to know. Your DNA double helix would unwind. The hydrogen bonds between the bases would break. And each of the strands are used as templates by DNA polymerase to add on complementary nucleotides to form the two daughter cells. And then you can go into a little bit more detail about the leading and lagging strands and about the replication bubble. Okay, I think that's enough for this one. Marram grass Ammophila arenaria is a xerophyte adapted to grow in sandy soils in exposed locations. Figure 4.1 is a light micrograph of a section through a leaf of marram grass. Okay, so we can see already immediately a lot of surface area on the spaces in the airspace near the stomata. A. arenaria is adapted to reduce transpiration state. What is meant by the term transpiration? Again, this is clearly a recall question. So the loss of water vapor from the surface of the leaves, and then you can talk about the whole sort of cohesion, adhesion, the movement through the stomata, and the way water would move into the roots from the soil. And also the process of how you get water vapor within the leaf. So the fact that liquid water evaporates from the surface of the spongy mesophyll cells. Okay. One adaptation of A. arenaria is the curled leaf. Give one adaptation, visible in figure 4.1, and explain how this reduces transpiration. Okay, so one of the easiest ones that you can say, cuticle, the waxy cuticle from all land plants. So the cuticle, the cuticle is, because it's waxy, it's impermeable to water. So it acts as a hydrophobic barrier to the loss of water, so the movement of water vapor in general, but this means that it will then reduce the loss of water vapor. You can even mention the fact that the cuticle helps reflect more sunlight, so you get less warming up over the course of the day. So that means less evaporation again. And you can also say that, so there's one of the most obvious ones, because I think the cuticle is kind of hard to see on this one anyway. But again, as we mentioned before, this sort of structure of the folded inner surface means that it can sort of trap the water vapor. So because there's more area here inside for the water vapor to be, the water vapor has to go through, has to diffuse through, a longer distance to be lost. So you reduce the rate of water vapor loss over time. Okay. And, okay. Yeah, I think that's good for that now. A student investigated the rates of transpiration and absorption of water by two species of plants, P and Q, over an 18-hour period between 6 a.m. and 24 hours. The environmental conditions for the two species were the same. Results are shown in figure 4.2. Okay, so we can see that P overall has a much higher rate of transpiration and water uptake over the course of the day than Q. But then by the evening, as the stomata close, the transpiration rate and the water uptake of P converges on to Q. Okay. Use figure 4.2 to calculate the difference between the rates of transpiration and water absorption for species P at time 1400. Okay, so we're looking at this value and this value. Yeah, okay. So that's exactly 60 minus, if we read this off, 42 equals 18. And be careful, what are your units? Per unit mass, so grams per hour. Okay. Describe and explain the patterns of transpiration and water absorption for species P. So you can see that over the course of the day, there is a dramatic increase in both transpiration and water uptake, and then a dramatic drop towards the evening. And you can also see that there is a time delay between the peaks of the transpiration and the water uptake peaks. And you also can see that there is, at this point, the water absorption becomes higher than the transpiration rate. Okay, and to explain this behavior, This clearly suggests that there is stomatal opening and closure throughout the day, hence the quick decrease in both transpiration rate and water uptake. And you can also suggest that you would get a higher rate of transpiration than species Q if there was a higher rate of sunlight, right? But in this case, that's not really that important, because we were told that both of these plants were under the same conditions. So this is more limited to the behavior of the stomata. Okay. And then you can sort of go into a little bit more detail about the fact that how these two values relate to one another. You can talk about the cohesion of the water molecules inside the plant, and how when a water molecule evaporates and then leaves the surface of the leaf, that will sort of move the water column along and result in the entry of water into the root. Okay. Suggest why the pattern of transpiration for species Q is not the same as for species P. So from this data, the fact that the peak is much lower and there isn't much difference between the transpiration rate in the evening or throughout the day even, this is a clear example of xerophyte. So a plant that is adapted to an environment in which there's little liquid water, like a desert or an ice snow covered region. And they are adapted to high light intensity during the day, which would be really dangerous if you didn't have any water, and you're way more likely to lose even that precious water that you have. So this is why they have their stomatal peak opening times later in the day than species Q. So species Q can afford to lose water. It's probably living in an environment in which water is more abundant, so doesn't have to worry about that. Okay. State the name of the causative organism of measles. So once again, this is clearly a recall question. So the morbillivirus would be the causative agent of measles. And then describe how the measles pathogen is transmitted. So measles is an airborne disease. So you can mention about aerosols. Aerosols are a colloid of fine particles of either a solid or a liquid in air or any other gas. So that happens when someone sneezes or just talks to you or just exhales. And then that colloid is then breathed in by someone else. And that's how you get the transmission. Okay? The measles pathogen contains a single-stranded RNA molecule and no DNA. The cells that the measles pathogen infects contain double-stranded DNA molecules. State two other ways in which the RNA in the measles pathogen differs from the DNA in the infected cells. Once again, very clearly a recall question. So the obvious ones that you can mention, so ribonucleic acid instead of deoxyribonucleic acid. So you have a ribose sugar instead of a deoxyribose sugar. You have the RNA bases uracil instead of thymine. And also you don't get the characteristic double helical structure that you would in the DNA. Okay? The measles pathogen must carry out RNA replication to make new RNA molecules for the new pathogens. This happens inside the infected cell. The pathogen carries its own enzyme for RNA replication, but no other enzymes. Explain why the measles pathogen cannot use an enzyme from the cell to carry out RNA replication. Okay, so our cells survive and proliferate by passing on genetic information in the form of DNA. Therefore, they don't have any sort of machinery for RNA replication. And also, we do have an RNA polymerase, but that uses a DNA strand as a template, not an RNA strand. So the cell has no machinery to carry out its replication. So therefore, it brings its own nice little encoded RNA polymerase. Okay, the outer part of the measles pathogen contains an antigen called the H protein. This antigen appears on the surface of cells infected with the measles pathogen. Describe the role played by T lymphocytes in a primary immune response to an infection by the measles pathogen. So, this one, once again, sorry, is a recall question. So you probably need to mention the fact that the immune system recognizes the presence of an antigen, which are presented to the immune cells by antigen-presenting cells. And then the T helper cells are T helper and T clear cells are recruited. T helper cells secrete cytokines, which then stimulate specific B lymphocytes, emphasis on specific, that will then mean that the plasma cells will release antibodies, and furthermore, it also stimulates the T killer response, where you can go into a little bit more detail on how that happens. So granzymes, you can mention the cytotoxic perforins that induce an apoptotic response, which means programmed cell death response in the pathogen. Okay, and also you can mention, obviously, so if you can mention just a little bit on the secondary immune response, so the fact that when you have the primary immune response, you also eventually produce memory cells, which then help a more effective immune response should an infection happen again in the future. Okay. Finally, figure 6.1 is a food web. From the food web in figure 6.1, name the producer. So the producer is the organism at the bottom of a food chain, which produces all the nutrients and the source of all the nutrients and food for the entirety of the food chain. So those are normally autotrophic organisms like plants. So that would be the grass. The organism feeding only as a primary consumer. So primary consumer means that they feed on only the producers. So from this, we can see rabbits are one, grasshoppers are also one, but mice aren't one because they do feed on grass, but also they eat grasshoppers. So that wouldn't be a primary consumer. Okay, the hawks are top predators. Name one other top predator shown in figure 6.1. So those would be the animals at the top of the food chain. So another one you can mention are the foxes. Okay, name the processes in the nitrogen cycle in which the following changes occur. This one, clear recall. I'm not going to draw the nitrogen cycle just not to make it more difficult, but nitrate ions to nitrogen gas. That happens by denitrifying bacteria in the soil, and then they get released into the atmosphere as nitrogen gas. Ammonium ions into nitrate ions. So once the ammonification happened, then nitrifying bacteria will carry out the conversion from ammonium to nitrate. And finally, nitrogen gas to ammonium ions. So these would be are symbionts, the nitrogen fixating bacteria that help plants access nitrogen gas from the atmosphere. And that's it. So thank you for everyone who's tuned in to this quick overview, and hope to see you again soon.